You'll remember that point mutations are defined as a change in a single base of the DNA. There are different categories of point mutations depending on the effect that the mutation has on the amino acid sequence found in the protein. And those different types are silent, missense, nonsense. Insertions and deletions we'll talk about in just a few moments, but let's come back and look at the first three types of point mutations. Again, silent mutations, missense mutations, and nonsense mutations. So as its name applies, a silent mutation is, quote, silent in the sense that it doesn't have a change on the amino acid composition of the protein. In this particular case, when they say they're showing the wild type DNA, they're showing the non-template strand. If we were looking at the double-stranded DNA sequence, there would be a T, C, G, C, A, and so on. The non-template strand will have the same sequence as the mRNA, except where there's a T in the DNA, there's a U. So please make sure you understand this is not the template strand. This is the non-template strand, and they're simply including it so that you can actually see the relationship between the DNA and the sequence of the mRNA that's produced via transcription. In this particular case, the mutation is happening to this particular base and it's being changed from an A to a T. Now, what impact will that have on the RNA? If there's an A on the non-template strand, then there's an A here. If there's a T on the non-template strand, remember there's a U in the uracil. Again, non-template strand of DNA sequence being shown. So what impact will that change have? Well, you can see that it has changed the sequence of the codon. So the second codon on the normal DNA is on the normal mRNA is GUA. Here we can see that changes to GUU. So you would go to the table of the genetic code, which we used in a previous topic, and find out which amino acid is associated with each of those. And if you look, you'll see GUA and GUU code for the same amino acid, and that's valine. So if you were to look at simply the protein sequence of amino acids, you would have no idea that a mutation had occurred in the DNA. A missense mutation means that a different amino acid is inserted as a result of the mutation. So in this particular case, you can see that we have a C, and that C found on the non-template strand is now an A. So how does that change the mRNA and the subsequent protein? Well, you can see that in this particular codon, instead of having CCC, which designates proline, there would now be ACC. And if you look up this codon on the table of the genetic code, the associated amino acid is threonine. So the sequence of amino acids has been changed. That means it's a missense mutation. A nonsense mutation means that a stop codon is inserted as a result of the point mutation. So we can see in this particular case, the last base is what's being changed from a C to a G. That means the last codon, instead of being UAC, which codes for tyrosine, it will now be UAG. And this is one of the three stop codons that you saw in the table of the genetic code. So the effect on the protein will be premature termination of translation. Normally, it would read through, add this amino acid, and then if the mRNA is longer, it would keep on going. But in this case, we would have, again, premature translation termination. The last two types of point mutations are insertions or deletions. And both of these have the effect of creating what's called a frame shift. So when we think of the word frame, we think reading frame, which is the orders of nucleotides contained in the codons on the mRNA. If there's an insertion or a deletion, then that means that the codon composition will change. So, for example, we can see in the original DNA, the sequence on the non template strand is AGCGTACCC, and after the mutation, the T and the A were removed. So when the mRNA is produced, remember the codon is composed of three consecutive nucleotides. So there would be AGC, AGC. So there's no difference there. But now we would have CUA on the original. There would be GCC on the new, and this represents a different amino acid. So you can see whether it's an insertion or a deletion that most typically is going to shift the reading frame and result in a completely different makeup of amino acids past the point of the insertion or the deletion. Now, how can the impact of insertions or deletions be minimized? It's if either three bases or a multiple of three are inserted within frame. So, for example, if there was an insertion of three bases after the first codon, then that would simply result in the addition of an amino acid between serine and valine. Now, that additional amino acid may have a devastating effect or it may have minimal effect. There's really no way to predict. But if you have an insertion or deletion of three bases or multiples of three that occur within the reading frame, meaning they don't cause a frame shift, they simply add or remove an amino acid, then the consequences to the protein are not as devastating, at least in terms of the amino acid composition. However, sometimes one change is all it takes to make a particular protein non-functional. So I hope this discussion is helpful, and there'll be an opportunity for you to practice using the self-assessment worksheet.